get started now. Hello, everyone. My name is Aline, and on behalf of Parkinson Society, I'd like to welcome you all to our webinar presentation today with Dr. Anurag Tandon. He's the Associate Professor at the Department of Medicine at the University of Toronto. The presentation will be approximately 45 minutes. Should you have any questions, just type them up in the text box that should be on the right-hand side of your screen. I will now pass it over to Dr. Tandon.
Thank you very much. Can you hear me well?
Yes, I can.
Okay, good. So I'm going to be talking about the intercellular alpha-synuclein transfer in Parkinson's disease. And it's kind of a complicated title, and I will start off with a little bit of background about Parkinson's disease. I think most people are, given that you are visiting this seminar, you're probably aware of most of the basic facts about Parkinson's disease. It was a disorder that was initially characterized by James Parkinson in the late 17 and early 1800s. And he actually wrote a quite interesting essay on this, and there's a picture of it here. And it wasn't until much later that the name Parkinson's disease was actually coined by another scientist named Jean-Martin Charcot. And the original name for PD, as James Parkinson named it, was the shaking palsy. And he didn't actually have patients himself. He would observe individuals in a old age home on the way to work. And he was a bit of a Renaissance man himself and had a lot of interests. And began this study that we are now pursuing quite thoroughly, both clinically, biochemically, and physiologically. And I'm going to talk a little bit about some of these features of Parkinson's disease that we understand, and some of the things that we don't understand quite so well. First, as you're all aware, the clinical features is age-dependent prevalence, and it's generally present pops up in a population that's older than 65, although there's quite a large incidence of the disease in a younger population. The major movement disorder that it presents with is a resting tremor, rigidity, bradykinesia, and postural instability. And these are all responsive to dopamine therapy, in particular L-DOPA. In about half of the cases of Parkinson's disease, you also get depression and dementia. And these are not treated well with L-DOPA, which suggests that they may arise from pathways that are non-dopaminergic in origin. Now, when we look at the pathology, there's a nice image here of a Lewy body, and you see the loss of pigmented dopaminergic neurons in the pars compacta of the substantia nigra, and shown here. And you also get these Lewy body structures. These are dense intercytoplasmic inclusions, and they are highly there's the protein that's highly abundant in these. It's called alpha-synuclein, and this is the protein I'm really going to be talking about a lot today. So the name alpha-synuclein was coined from a study in the electric fish, and it's a protein that's highly abundant in nerve terminal, and it turns out also in mammals. We find a lot of it in structures that neurons use to communicate with each other. The function and the behavior and the properties of alpha-synuclein are really less well understood, but over the last 10 to 15 years, we've really begun to dissect out some of the features of alpha-synuclein that are important for its function, and in particular that are important for the development of Parkinson's disease. I also want to mention a couple other things, and we always think of Parkinson's disease as a dopaminergic disorder, and that's related to changes in the substantia nigra, and those neurons in the substantia nigra project to the striatum. However, there's quite a bit of other pathology that's also apparent in PD, and that's there's a involvement of the cerebral cortex, particularly the brainstem. The autonomic nervous system is also involved, and the autonomic nervous system, which controls things like heart rate, gastric motility, the dilation of the pupils, salivation, all of those things are really important for daily living. And there are some aspects of this autonomic nervous system which are affected in Parkinson's disease, and it seems that they might be actually the first site of Parkinson's disease as a clinical feature. So these are quite important, and I'm going to talk a lot about those today. And some of the pathways, we talk about Parkinson's disease as a dopaminergic disorder, but if you look at the other neurotransmitters such as cholinergic, noradrenergic, and serotonergic pathways are also affected. So I would just like to stress that although the movement phenotype of Parkinson's disease, which can be corrected by dopamine therapy, a lot of the other aspects that are apparent in PD are not corrected well with dopaminergic therapies, and we need to look for additional approaches in how to correct those. Now, I'm going to come to the idea that Lewy bodies are an important aspect of Parkinson's disease, not just because they are a very classic feature of the disorder, but they may also be involved in propagating the disease. So these Lewy bodies, shown here, are primarily very sticky deposits found inside neurons, and they are comprised of large amounts of alpha-synuclein and a number of other proteins such as ubiquitin neurofilaments, also a number of trace metal elements. Now, these Lewy bodies, and synuclein in particular, is not specific to Parkinson's disease. And there is a broader group of diseases that we have now collectively termed synucleinopathies because of the aggregated synuclein that's present. So this includes Parkinson's disease, Alzheimer with Lewy bodies, dementia with Lewy bodies, multiple system atrophy, and some more obscure ones like Halliburton Spatz disease, which is now called neurodegeneration with brain iron accumulation. It's a mouthful. And those diseases most likely have a very similar etiology. In other words, how they develop may be very similar to Parkinson's disease, but the areas that are affected are quite different. So I'm going to focus primarily on PD today, and the next section I'm going to talk about is trying to understand how this Parkinson pathology develops over time. There was a major advance recently, and there were a couple of studies that suggested that Parkinson's disease pathology may be able to spread from one affected cell to another. And the way this came about was two groups, Jeffrey Kordauer's and Patrick Brundtland's groups, in collaboration with people like Warren Olenow, also found that they looked at postmortem brains of individuals who had Parkinson's disease and then had received dopaminergic grafts. And at the time this was done, these patients prior to death, had received these grafts somewhere between 8 to 15 years prior, and some had unilateral grafts, others had bilateral, so one side or both sides of the brain had received these grafts. And what they looked for upon postmortem examination, they looked at the substantia nigra of these individuals, and sure enough, as they expected, found that there was quite a few Lewy bodies present. What was really surprising was that they found that in the graft tissue, they also found neurons with developing Lewy bodies. This was completely unexpected because the graft tissue, if you recall, was all fetal tissue. In other words, it was very young. So at the time of this postmortem examination, the graft tissue would have been only 8 to 15 years old, whereas the Parkinson tissue, substantia nigra, or the patient's tissue, would obviously have been well over 65 years. So unexpectedly, they see these Lewy bodies in what they would have thought to be healthy tissue, and this raised a possibility that the conditions of the brain of these Parkinson patients is actually causing a change in the healthy tissue. In other words, something from the host tissue is being passed on to the grafted tissue and causing Lewy bodies to appear. And this has raised a completely new paradigm in Parkinson's disease, in that we think now that the Parkinson pathology may be transmissible from one cell to another. So on this cartoon, the non-transmissible form, you have a neuron that develops Lewy bodies, and this is due to whether some sort of inherent mutation in its genes, or it may be an inherent insult that it receives or under oxidative stress. Nevertheless, this neuron develops a Lewy body, but then doesn't pass it on to this. The other neurons are, if they do develop Lewy bodies, they would have to do so independently. In this new paradigm, you have cells that develop a Lewy body, then pass it to the next, and on and on. And this allows that pathology, therefore, to spread from one cell to another if they're in nearby, either in contact or very close together. And this, of course, is an aspect of the disease then that would really modify how we think about treating it. In other words, if you think about this, we've got to be able to try and block the protein aggregation, the Lewy body formation inside a cell, which is much harder than trying to block the delivery from one cell to another. And therapeutically, it would be much easier to try and block these cell-to-cell connections and try and block proteins being moved from one cell to another, or whatever that diffusible messenger is. Now, prior to these studies, this hypothesis that there's a temporal and topographic progression of Parkinson pathology had been discussed much earlier, and that was by a group in Germany, Heiko Braak and his colleagues, and it has now come to be called the Braak Hypothesis. And what that group did was looked at Parkinson brains and then tried to linearly place the pathology in order of some sort of progression. And so what they came up with a scheme that has stage 1 to stage 6 in terms of how the disease might develop. In stage 1, they considered this to be the preclinical phase of the disorder. And they noted that Lewy bodies were present in the brainstem in a variety of different areas, both in the brainstem and in an area called the olfactory bulb. But these patients, of course, are not presenting with Parkinson's disease because they would be considered preclinical. It was only stage 3, what they called the Parkinson's disease, in which there was a motor component to these individuals. And by that point was a change in the progression of Lewy bodies into the substantia nigra. So, of course, the first point in the disease where a movement disorder appears is when the substantia nigra is affected. And going further, individuals would then develop these Lewy bodies further up into the cortical areas, and this could account for some of the later symptoms of Parkinson's that we see, for example, the depression and the dementia. Now, there are ways of looking at how these different stages might correspond to the both motor and premotor symptoms that we see in Parkinson's disease. But I want to bring your attention to two particular parts of the brain that are, I think, that are important in how we dissect out the initial stages of the disease. And the first one's called the dorsal motor nucleus. This is of the vagus. With the vagus is a peripheral nerve that controls a number of different features of your normal living, particularly things like heart rate. It'll also control gastric motility. And the cells of the vagus are located in this particular nucleus of the brain called the dorsal motor nucleus. And this is located in the brainstem, fairly low down in the brainstem. There's also changes in the anterior olfactory nucleus, which is located here. And I'm going to come back to this and explain the connection between these as the initial site of pathology. So both of these aspects of the progression of Parkinson's disease, as well as the concept that synuclein might be transferred from one part or another part of the brain, or maybe a diffusible messenger in those studies where they looked at the graft tissue in developing Lewy bodies. This has now brought about a new hypothesis in Parkinson's disease, which is called the Prion hypothesis. And prions are small infectious proteins, and this has really come about from the idea of prion disorders such as mad cow disease, which there are variants also present in humans. And although we know that Parkinson's disease doesn't spread from one person to another, the hypothesis has nevertheless been proposed that synuclein may be an infectious particle that can spread from cell to cell. So if you look at this neuron, you've got normal components of the cell, and these sort of rainbow colored structures I've drawn here are alpha-synuclein in their normal stage. Now, something might happen to the cell that causes these molecules to now change and become misfolded in some way. So this could be some sort of unknown genetic or environmental trigger. And this continues on until all the synuclein, or most of the synuclein, has now become modified. This involves a propagation of alpha-synuclein pathology that involves seeding and recruiting of all the additional synuclein. And ordinarily what would happen is the cell would now either die because it doesn't like this misfolded protein, or it would try and compartmentalize these proteins. And this is what we think that Lewy bodies do, in fact. They protect the cell by taking these misfolded proteins and compartmentalizing them into one part of the cell that then protects the cell from dying. Now, under certain conditions now, what might happen is that an adjacent cell nearby, which would also be healthy, would have normal synuclein. However, through neuron-to-neuron transfer, we get this propagation of synuclein misfolding, which now converts the synuclein in the new cell into this sort of unhealthy and diseased type of synuclein. And of course, the Prion hypothesis would then state that this cell now would be able to connect to another neuron along the chain and then continue the process and the propagation of this pathology. Now, we can look at some of these aspects by looking back towards the preclinical non-motor symptoms of PD. So the question we can ask is, when does Parkinson's disease actually begin? And we can measure neuropathological and PET imaging changes largely 5 years prior to a person showing up at the clinic with a movement phenotype. Now, in a study that was done in Honolulu and Asia, they looked at middle-aged men over many years, and they found that individuals who developed Lewy bodies were much more likely to have complained about constipation many years earlier. Now, this was a somewhat unexpected finding, suggesting that Parkinson's disease had a peripheral or a gastric component. It really hadn't been anticipated at that time. But what they found, and this is quite striking, is that there was Lewy body pathology in 25% of males who had constipation versus 6.5% without. They also had fewer nigral neurons in this cohort that had complained about constipation. And these results have also been replicated in a female study. And more recently, a couple of studies have shown that colon biopsies of patients with early PD do show alpha-synuclein pathology. So this is quite an interesting aspect of the disorder, which was completely unanticipated. We have anxiety disorders that appear 20 years earlier, also some rapid-eye sleep movements, sleep behavior disorders that can occur 15 to 20 years earlier. Anemia, which is a bit more of a vague finding, but there's evidence that there are mitochondrial changes in individuals with PD, particularly as related to blood cells. And lastly, I just want to highlight this particular one, which is olfactory impairment that can be seen in individuals 2 to 5 years earlier. And these two aspects, constipation and olfactory impairment, are both really important when we think about the aspects of the Lewy body pathology at the very early stages of the disease. Now, as I mentioned, the dorsal motor nucleus of the vagus, which is one of the earliest sites of Lewy body pathology, connects to the gastric tissue and actually controls gastric motility. And this could account for changes in gastric motility in these patients or in these individuals prior to them developing Parkinson's disease. Similarly, the Lewy body present in the olfactory bulb, which is involved in collating sensory signals from the nasal passages and into the olfactory nerves, and then sending them up to the cortex, could account for the loss of olfactory function prior to the movement disorder. So what is the connection between the olfactory and the digestive tracts? Now we know that these are linked because, well, you have pathogen entry. It might occur either through nasal passages, so the mucus is then swallowed and taken into the stomach or beyond. There's also the possibility that the toxins may be swallowed similarly, and both of these would now affect the peripheral nerves that regulate olfaction and gastric motility. The nerves that connect these connect to the olfactory bulb and the dorsal motor nucleus of the vagus that's located in the CNS. And I'm going to show just a couple of slides that demonstrate just how close the olfactory nerve, for example, the olfactory bulb and the olfactory nerves are to the nasal passages. It's actually only a matter of millimeters. So one can imagine environmental toxins or viruses that might be able to enter. Now, when I mention environmental toxins or viruses, this is purely speculation. It's really unknown what the initial triggers are, and one presumes that there may be some sort of genetic component to the susceptibility to these particular toxins. Nevertheless, one can imagine that toxins passing through this short nervous tissue into the brain could affect alpha-synuclein aggregation in the olfactory bulb. And if you look at this section, which is a view of the brain from the bottom up, you see the olfactory bulb right here, and also the vagus that exits the brain from here. And these two areas, now showing in these pulsating circles, are connected to the initial site of pathology. So the dorsal motor nucleus is located right here in purple. This projects out to the gastric tissue, and it actually does contain quite a large amount of alpha-synuclein in these neurons. And I'm going to go to the next slide, which shows you how the connection might occur. So this is a section of the brainstem with the dorsal motor nucleus on each side, labeled in the X on this side. This projects the gastric branch of the vagus, and that controls the muscles around the stomach lining and the intestinal tract. And this control is generally not under conscious control. It's what we call the autonomic nervous system, and when you eat, it becomes more active. When you're in conditions where you're doing exercise, for example, or in what we call the fear or flight mode, these become less active. Now, if you look at this, the dorsal motor nucleus in postmortem analysis, there's quite a small area that, when magnified, you can see the appearance of Lewy bodies in Parkinson's disease, and also the accumulation of alpha-synuclein in what looks like Lewy bodies. So very similar to what we would see in the substantia nigra at later stages of the disease. Now, As I mentioned, a couple of studies recently also looked at the appearance of colonic alpha-synuclein labeling. And this is really important because the goal of these studies was to establish whether we could detect alpha-synuclein pathology with a very simple colon biopsy that could be done on a walk-in basis to your doctor, where they take out a small piece of tissue and then gets examined pathologically and might determine whether you have or might be on the way to developing some sort of alpha-synuclein pathology. Now, in these cases, they had this particular study done by Cordauer's group, Shannon and Al, was published about 2 years ago, and they looked at 10 Parkinson cases and a number of controls. And they were lucky enough to get patients that were very early in the treatment that had had colon biopsies for some other reason, and were able to track down the biopsy tissue and look at synuclein labeling. And what they found in these control tissues was virtually no labeling of alpha-synuclein at all. And when they looked at the Parkinson cases, they established that there was significant alpha-synuclein labeling or pathology that was present in all of the Parkinson cases. So it was quite a striking finding. And subsequent studies, this one, Gold et al., which was actually done here in Toronto by David Munoz's group at St. Mike's, what they found was that they replicate part of this to the extent that they also found that 100% of the Parkinson cases were positive for alpha-synuclein pathology in the colon. However, only about 50% of the general population was free of synuclein pathology. And this was the same whether there were 80 patients as well. So that there appears to be some specificity of the synuclein pathology in the enteric tissue relative to Parkinson's disease, although it wasn't quite as striking as the original finding. Again, both of these studies had fairly small numbers of patients, and they really do have to be replicated in a broader study to understand whether this can be treated or considered as a real biomarker or some sort of signal to determine whether someone might be developing Parkinson's disease. It is a very exciting finding, but I think we're still at the early stages to try and understand whether there are multiple types of pathologies. In other words, do the 50% of the general population that show Parkinson's disease like pathology, are they really going to be developing PD, or is there some thing that we are missing, that there's something different about the general population, that although they do have some sort of alpha-synuclein staining, it may be different from the PD population. Nevertheless, this is something that I think is going to be really heavily pursued in the next few years. Just as a side, we've been working on these kind of studies as well in my group, but we're using animal models, and in this case, when we look at our transgenic animals, once that study with the colon biopsies came out, we actually looked at our animals to see whether they had synuclein expression in the enteric neurons. And sure enough, you can see here, this is in our transgenic animals that are basically bred to have more than normal levels of human synuclein. So these animals have no mouse synuclein, but they express human synuclein. And you can see that there's quite a bit in these neurons. These are neurons that are on the outside of the enteric or your stomach lining. In non-transgenic animals, there is still some staining, but it's much fainter. And this is what we would consider the normal level of mouse alpha-synuclein in the gut. And these are just two other pictures showing co-labeling of alpha-synuclein with neuronal markers. So we know that where we see alpha-synuclein staining in the stomach lining is they are really neurons and not some other sort of tissue like such as muscle tissue, which you can see here in blue. So what do we know about alpha-synuclein? This is a protein that my lab has been working on for many years, and we're really interested in this. And our focus is, for the most part, been to try and understand what alpha-synuclein does under normal conditions. The reason we were interested in alpha-synuclein because it's a protein that's found heavily in brain. There's a lot of alpha-synuclein. It's also highly present in the parts of the neurons that are involved in communicating from cell to cell. Its role in Parkinson's disease was actually only discovered in 1995. So although we knew about alpha-synuclein as a protein involved in neuronal transmission much earlier, it was identified as a key factor in Parkinson's disease. It was the first gene to be shown to be involved in PD, and subsequently a number of others that were identified. I'm going to just give a short classification scheme of the different Parkinson's genes, and I like to put them into this sort of scheme because it helps me understand what their function might be. And I put both genes, LRRK2 and alpha-synuclein. These are both autosomal dominant genes that you need a single mutation to cause a disorder. Both of these are involved in what we call neuronal function. Alpha-synuclein, so here's a neuron with a cell body, an axon, and a bunch of projections at the end, which we call nerve terminals. These are where we think alpha-synuclein functions. It is highly abundant in these structures and is involved in the communication of this neuron with other ones along here. Now, LRRK2 seems to function more at the cell body, where it controls the arborization of the dendrites, these are what we consider the receptive areas of the cell, which receives information from other neurons. And we have a number of proteins or genes that are involved in protein degradation pathways, such as proteasomes or autophagosomes, lysosomes. And lastly, there are some that are involved in mitochondrial function, and this is really important in Parkinson's disease, understanding how energy metabolism occurs. And so some of the autosomal recessive genes, such as PINK1, DJ1, and Parkin, seem to be involved in regulating how mitochondria behave and produce energy. And some recent studies have also implicated alpha-synuclein as being involved in this pathway. However, regardless of which gene and which pathway is involved in causing the PD, we know that alpha-synuclein is one of the endpoints. And for this reason, I like to call alpha-synuclein a cause and endpoint of Parkinson's disease. That's because we know that we always get an accumulation of this insoluble alpha-synuclein in Lewy bodies, as shown here. This is Parkinson brain substantia nigra, which shows Lewy bodies as well as dense aggregations of alpha-synuclein staining. And there's also a second protein, which is heavily found in these Lewy bodies, which is ubiquitin. And it's usually found in conjunction with alpha-synuclein. So what do we know about the alpha-synuclein gene? We know that there are 3 mutations in alpha-synuclein that cause PD. As well, if you overexpress if you overexpress alpha-synuclein in humans, it seems to be causing Parkinson's disease, because we know that gene multiplication, either duplication or triplication of the normal alpha-synuclein gene, can also predispose individuals to PD. Now, the molecule, this sort of a schematic of the alpha-synuclein molecule is shown here. It's got 140 amino acids. This is what we call the N-terminal, or the beginning of the molecule. This is the C-terminal, or the tail end of the molecule. Now, if you notice, the mutations are present in the first half of the molecule. And this area has a certain series of repeat sequences. And these repeat sequences are important for how alpha-synuclein behaves. This is what we call a membrane, or a vesicle in nerve terminals. And alpha-synuclein is able to fold such that these repeat sequences allow it to coil up. And this is called an alpha helix coil. And what this coil does, it aligns a certain number of those amino acids on one side that seem to like lipids or fats, And those on the other side that like water, which is the aqueous medium in the cell, and so it allows alpha-synuclein in this conformation called an alpha helix to bind to lipid membranes. And then there's this C-terminal or tail end, which really is like a tail in this molecule, that sticks out and has some sort of unknown function. This tail also gets another modification, which is called a phosphorylation. And this phosphorylation in normal tissue is only on about 5% of synuclein molecules. However, when you look at the synuclein that's present in Lewy bodies, about 95 to 100% of that synuclein is phosphorylated. That has this particular biochemical modification. And this suggests to us that this particular modification is important for modulating the structure or the function of this tail sequence. Now, if you look at Lewy bodies that are isolated from Parkinson brain, they have these really amazing radiating fibrils. And when you take these fibrils and isolate them biochemically, there's a structure shown here. You can see these fibrils, and using antibodies that are labeled with gold particles, the antibodies will decorate these fibrils. And because we know that antibodies that are targeted to the C-terminal tail, as shown here in these ones, decorate all the way up along top to bottom of the fibril, we know that C-terminal tail is sticking out. If you now take antibodies that are targeted only to the N-terminal or the head of the first half of the molecule, they can only pick up one epitope, and that's right here. What this tells us that the amino-terminal of the beginning half of the molecule is actually buried within the structure, within the fibril, and cannot be accessed by the antibody, whereas the C-terminal domain sticks out. So if you look at this structure, this is how I would explain this. The amino terminal is present here. The first half of the molecule forms this dense structure that forms a backbone of these fibrils, and then the C-terminal tail sticks out, and that one can still be accessed by various antibodies, whereas the amino terminal or the head of the molecule is only present at one end. And what's really interesting about this is that amino terminal half, or the first half of the molecule where the mutations are, is also important for membrane binding. So the same sequence which is involved in membrane binding is involved also in forming this fibril structure. And we know that there is some sort of structural change, and I'm going to refer to it as misfolding of alpha-synuclein that goes from this alpha helical structure to this fibril structure, which we refer to as beta sheet. So if you ever hear of someone talking about beta sheet, you know that it means this sort of odd, dense fibril structure of alpha-synuclein. Beta sheet can also refer to other proteins such as amyloid protein in Parkinson's disease, which also forms this sort of beta sheet structure. So if you look at alpha-synuclein in healthy tissue, we know that it exists in this alpha helical structure bound to various types of membranes, shown here in yellow with the C-terminal tail sticking out. And this membrane-bound alpha-synuclein is in equilibrium with a soluble diffusible alpha-synuclein, which is floating around inside the cell and is very flexible, shown here in a very pleasant rainbow color. Now, under certain conditions, this alpha-synuclein can now form that beta sheet structure, as I mentioned in the previous slide, and start beginning to form these fibrils. This may occur in particular with various types of oxidative stress and other types of modifications to alpha-synuclein, and I'll come back to that. As I mentioned, that particular biochemical change of the C-terminal tail, which is the phosphorylation, could also cause a change in this structure. Now, if one were to think about this self-assembly of alpha-synuclein, we can see that as this condition occurs and progresses, these fibrils will become longer as more molecules of alpha-synuclein are added on, and then we presume, although we don't fully understand how this goes to these Lewy body structures, but we can assume that the cell now tries to get rid of these and begins to compartmentalize them in a one localized part of the brain, or sorry, part of the cell, much like we do with a landfill or garbage dump. That the cell is really trying to get rid of this and put it out of the way so that doesn't bother its normal functions. Now, as I mentioned, this phosphorylation change, or this modification of the C-terminal, is heavily seen in these Lewy body structures. And we've been involved in making some of these Lewy bodies fibrils with phosphorylation. And I'll come back to them in a few minutes. But when you look at synuclein in the normal tissue, or normal brain, so here's a schematic on the right side that has a neuron with a cell body, an axon, and a nerve terminal. And really it's this nerve terminal area where synuclein is really, really prevalent. And it's involved in transmitter release, which is encased in these small vesicle-like structures. So there's transmitter stored inside these, and when a neuron is stimulated, it releases this transmitter into the extracellular space where a receptive neuron will pick it up and communicate it on further. So this signal is the underlying feature of how neurons communicate with each other. Now, when you look at these vesicular structures, as shown in this electron micrograph here, so these circular structures are what we store neurotransmitter in, and immunogold labeling, so these are antibodies that are tagged with a little gold particle that can be picked up on electron micrographs, can be seen here. So these are antibodies that are directed towards alpha-synuclein, and you can see that alpha-synuclein therefore is present on or very close to these little vesicular structures. And we do now understand that synuclein is an important component in regulating how neurotransmitter occurs. So, for example, if this was a dopaminergic nerve terminal, you would have this part of the neuron in the substantia nigra, and the axon, which then projects to the striatum, and then the dopamine nerve terminals present in the striatum. Now, under certain conditions, and we can do this in basically a culture dish or in a test tube. We can purify alpha-synuclein. So in this case, what we're showing you is alpha-synuclein that contains an A53T mutation. It's one of the Parkinson mutations. And when you just leave it, if you incubate this at 37 degrees shaking or in the lab, we can generate these lovely fibrils, so you can see here. Now, the question that still remains is whether these fibrils are the same thing as what's contained in here. Now, obviously, there are most likely some structural changes that we don't fully understand, but it is reasonable to assume that these are the type of fibrils that eventually form this Lewy body structure. And my lab has been involved recently in some studies to look at how these fibrils form. And what we find is that there's when I mentioned the phosphorylation change on the C-terminal tail, what we find is that when we generate synuclein molecules that have this particular phosphorylation, we get a very accelerated fibril formation. So here you can see normal, or what we call wild type alpha-synuclein. This is normal synuclein without mutations. And two types of synuclein would either have the A30P or A53T mutation. These are both mutations that are present in Parkinson's disease. And if we purify this synuclein, we can see that this is done 9 days incubation at 37 degrees. These begin to aggregate, but they don't form fibrils quite so fast. When we look at the phospho versions of these alpha-synuclein molecules, in other words, they've been phosphorylated at the C-terminal, and this is the one that's present in Lewy bodies, you see this incredible acceleration of fibril formation. We have now done a number of studies with these. If you take this fibrils and treat cells with this, and here what we're looking at is the uptake of this fibrillar alpha-synuclein. This is the A30P molecule by cells in culture. So the green here shows alpha-synuclein in these. In this case, these two are the non-phosphorylated, and these two are the phosphorylated alpha-synucleins treated in these cells for either 1 hour or 72 hours. And what you see is there's a small amount of accumulation in the non-phosphorylated. The green shows here, which accumulates in the cells. Over 72 hours is a lot more, obviously, and it's also internalized. When we look at the phosphosynuclein, there's a clear increase in the amount of phosphosynuclein being bound by these cells and accumulated. And over 3 days, 2 hours, we see quite a bit more alpha-synuclein, some of it being internalized, but significantly more than the non-phosphorylated. So we think that the phospho really modifies how synuclein aggregates, and how quickly it's taken up by cells. And here's another one which shows this same idea. Here, this is 3 days as well. This is non-phosphorylated alpha-synuclein here. You can see some punctate staining around on the inside and the outside of the cell, whereas when you look at the A30P phosphorylated version, you can see quite a lot of alpha-synuclein being taken up. And so what this is telling us is that the fibrils that we can generate in vitro or in a test tube, we can add to cells and see that it's actually taken up, which tends to support the idea that synuclein can move from one cell to another and cause changes in the accumulation of alpha-synuclein in a receptive cell. And I'm just going to end. This is my last piece of major data, and this was published several years ago, and this was a really interesting study. And what this is Virginia Lee's group in Philadelphia, and what they did was they looked at whether, much like we're doing with the cells, whether you could actually transmit alpha-synuclein pathology in a mouse brain by injecting fibrils. And so what they did was they injected, for example, up here shows the injection of alpha-synuclein misfolded protein aggregates or fibrils, and looked at the pathology over time, and were able to find that there were dense aggregations of alpha-synuclein appearing in the neurons where the injection occurred, but also at a really far away regions of the brain. So if the injection was done here, you could see, the injection that was done in the black square, you could see pathology appearing in all sorts of other regions of the brain. In other words, the ability of alpha-synuclein fibrils to transmit from one area of the brain to another area of the brain was validated in this particular study. If you inject these brains with just what's called saline, without any fibrils, you see that there's no propagation of aggregation, which suggests that it's not just the injection itself which is causing it, but in fact it is the addition of the fibrils to it. So this validated the idea that synuclein fibrils might be able to induce pathology when injected into the brain. So I'm going to try and wrap up a little bit of this here by explaining some of the different mechanisms that synuclein might be able to undergo release and then undergo uptake in another cell. There are several different pathways that this might occur, and this is getting quite specific at this point, but we don't really know which one of these pathways is really responsible for alpha-synuclein. The reason I bring this up is because these are the type of things that as a scientist, we would want to identify specific pathways that we could then modify or block under certain conditions. So there are these what's called a classical form of exocytosis. In other words, synuclein is incorporated into these vesicular structures, much like a neurotransmitter would. These structures then fuse with the cell membrane and release alpha-synuclein to the extracellular space. Now, this synuclein would then be able to traffic to a neighboring cell where it would bind to a specific receptor or onto the membrane and be internalized under these conditions into vesicles that are budding off the membrane. Similarly, there's the possibility that synuclein may actually be able to transfer directly through the membrane because this is a membrane-binding protein. It may actually be able to form small pores in the plasma membrane of nearby cells and then transport to the inside. Now, there's another form of alpha-synuclein release, and this is called exosomes, and this is quite interesting because what this allows alpha-synuclein to do, the cell is really trying to get rid of misfolded proteins using this particular method, and it takes misfolded proteins and then encapsulates them in membranes, which it then sort of chucks out. It's much like we would put our garbage bags out on the front of our driveway for pickup. Now, it's not actually quite as noble as that. It actually just sends them out, and if there was a garbage truck, it would be really great. But what happens is these encapsulated structures now travel to another neuron, and they can actually bind and then be taken up by those neurons. And this internalized synuclein can now be involved in aggregating or propagating its misfolded structure. So the things that we would like to target, of course, is blocking these type of structures from releasing alpha-synuclein, or being able to block this alpha-synuclein if it's in free form outside cells. And we know that synuclein is present in brain in the extracellular fluid, so cerebral spinal fluid has quite a bit of alpha-synuclein. We can see it in blood. So it does seem to be able to get out of cells. And we've known this for a long time. We've always assumed that this was because cells were dying and therefore they would release the synuclein. However, they may be specific mechanisms that we can now target to understand ways in which we can block the transfer of synuclein from one cell to another and thereby impede the transmission of pathology. So I want to just summarize this talk by just reiterating some of the things that I've mentioned. And the first is that Parkinson pathology may be initiated in the peripheral nervous system. The reason I say may is only because so far we don't have validation of the idea that peripheral fibrils or synuclein fibrils can cause, for example, in the gut, can cause a change in brain pathology. So this is something that is still being studied intensely, my lab included. We're looking at ways that in animal models to see whether synuclein pathology can be propagated from the gut into the brain. We do know, however, that the pathology seems to follow specific neural tracts, at least in the brain, by cell-to-cell transfer. And this was shown in that study where fibrils were injected directly into the brain, and subsequently that pathology then spread throughout the brain. So although we don't know whether there is a direct link from the peripheral nervous system of this pathology into the central nervous system, we certainly do know that once it's in the brain, it can certainly spread from one place to another. Now, the main component of this particular propagation is misfolded alpha-synuclein. That doesn't mean that it's the only component that is present in those fibrils. There may be differences in the shape of alpha-synuclein, that there may be other proteins that are associated with it. So this is something that's still important and worthwhile to understand. For example, is the synuclein much worse when it's phosphorylated, for example, or is that just a side issue? If in our lab we've seen much higher aggregation of alpha-synuclein when it's phosphorylated, which is the Lewy body type of alpha-synuclein, and that this particular phosphorylation also changes how it's accumulated within cells. And lastly, and most importantly, I would say that this information is we're going to develop therapies aimed at blocking alpha-synuclein misfolding and cell-to-cell transfer. I think these are probably the most useful ways in reducing the disease progression. What we'd really like to have is something that we can block, a treatment that we can block at any stage of the disease, obviously. But as in most cases, most diseases, the earlier we treat them, the better the treatment would work. And that, I think, in the case of any neurodegenerative disease, I think that will be a truism. And I'm going to end there, and I'll be happy to take any questions.
So I think there's a question on the right-hand side of your screen by Mr. Joseph D'Souza.
Okay. Let me see if I can, how do I get back to that? Oh. Joseph D'Souza is asking, is there any work done with alpha-synuclein transfer done in vivo within non-primate primates? There is, and I think this is really at the early stages right now. I think the difficulty with doing experiments with human primates is largely cost and the length of the experiment that they take. So it is much harder, and I agree that at some point these will have to be replicated in non-human primates. I don't think they are. There's been no publication of that yet, but there are groups that are using non-human primates primarily for looking at behavioral aspects. And those animals that have been treated and studied for behavior may at some point be assessed for post-mortem analysis. So I think your question is really valuable, particularly from the aspect of being able to reproduce the disorder in non-human primates and then being able to treat them.
Okay. Anyone else have any other questions? We have another one.
Does DBS, so does deep brain stimulation affect alpha-synuclein in any way? Wow, that's a good question. And the short answer to that is, I don't know. One of the problems, so deep brain stimulation is the insertion of electrodes into specific parts of the brain which allow the modification of signals, particularly in the basal ganglia, of causing unwanted movements. And I think the downside of that is you're looking at a very select circuitry, so that where pathology might be occurring is not necessarily where you're inserting the electrodes. The electrodes are really there to change the electrical signals from one part of the brain to another. So my guess is it probably wouldn't affect alpha-synuclein structure in any way, largely because it's an electrical signal, and I don't, to my understanding, there is no effect of electrical stimulation, at least in the normal neuronal sense that we have in the type of stimulation one would see of neurons in brain. They are unlikely to cause alpha-synuclein to aggregate. There is the possibility, however, that the stimulation causes the release of alpha-synuclein, much like, as I mentioned, a transmitter would. So in that sense, it's possible that synuclein might be released more than normal would. But I'm merely speculating at this point, and I would say that there is no evidence to date that DDS would affect alpha-synuclein in terms of its misfolding anyways.
Okay, does anyone else have any other questions?